Hello everyone, my name is Natalia López. I'm working with Carol Landines, Professor Carolina Montoya, and Professor Clara Saldamando. We are from Universidad Nacional de Colombia. Our project is called Spodoptera frugiperda virome core strain based on RNA-seq, identification of potential biocontrols. Okay, let me introduce you to the problem we are dealing with. Spodoptera frugiperda is a pest that causes severe damage to corn and rice crops in Colombia and leads to millions of dollars in economic losses worldwide. Its increasing resistance to insecticides makes the search for sustainable control strategies urgent. Among these, the use of microbiota and insect-associated viruses, such as baculoviruses, shows promise due to their stability and genetic manipulability. However, in Colombia, limited knowledge of Spodoptera frugiperda associated viruses restricts the development of innovative management strategies for this pest. This is why the objective for this project is characterize the virome of larvae Spodoptera frugiperda corn strain to identify viruses with biocontrol potential. In the methodology, 3 larvae of Spodoptera frugiperda instar 5 were collected in Envigado, Antioquia, Colombia. We sent the RNA extracted to be sequenced parent with the technology Illumina NovaSeq 6000 platform at City Genomics in New York. Here is the bioinformatic workflow, with each color representing a different step in the process along with the respective programs. The first step was data preprocessing, where we took the raw sequences and performed quality control and trimming using FastQC and Trimomatic. Next, we mapped the sequences against Spodoptera frugiperda reference genome to filter out host sequences from the analysis. With the unmapped sequences, we proceeded to perform de novo assembly using 2 different programs, Trinity and RNA viral spades. For taxonomic identification of potential viral sequences, we used BLAST-N and BLAST-X. It's worth to mention that we downloaded a viral-only database from NCBI for this analysis. And finally, to verify the identified viruses, we perform another megablast and BLAST-N against the NCBI non-redundant database. The transcripts that passed that verification are considered viral sequences. This is a work in progress, so we have preliminary results. The first one is about the quality analysis. Low quality nucleotides that could interfere with the analysis were removed. In the second graph, it indicates that after removing the initial positions, the base content appears generally balanced, which is a good indicator of quality. As I said before, we used 2 different de novo assembly programs, Trinity and RNA viral spades. We want to compare approaches. So Trinity optimizes complete transcriptome reconstruction, while RNA viral spades being designed for specifically viral sequences, generating more transcripts in virus-rich samples. Since our goal was virome analysis, RNA viral spades proved more sensitive for detecting viral diversity in the sample. So to continue with the analysis, we used the results, the transcripts generated by RNA viral spades. For the virus identification, I show you here the BLAST-N results. It was performed with this eValue against a reference virus database downloaded from NCBI. So initially, viral sequences from the baculovirus family in green and multinucleopolyretrovirus in yellow were found. Most of them with the identity percentage above 70% and alignment lengths over 100 nucleotides. but we wanted to verify the blast results by performing a megablast and against a non-redundant database. And unfortunately, we were surprised to find that the results from the two analyses didn't match at all. So in this part, in the green one, we show you here the results from the blast N against viral database, and it's a nucleopolydro virus, right? But in the orange one, the megablast against a non-redundant database, the results was completely different. Was transcripts of Spodoptera frugiperda. So there was an inconsistency there. We are currently analyzing what's going on with the results, what's the difference, maybe because of the sequencing parameters of the bioinformatic workflow. Also, the viral databases are usually more specific and may contain sequences that are more representative of certain viruses. But for the other hand, the non-redundant database, which includes a big range of sequences from various organisms like bacteria and so on, and maybe affect the results. So for the conclusions, for posterior analysis, it's necessary to test other transcriptome analysis programs, and it's essential to determine the cause of this inconsistency in the results in order to provide more reliable outcomes and conduct further analysis, because in this moment we can't say that we found that viruses. So if any of you work with this type of analysis or data, we would appreciate your feedback and suggestions. Thank you very much.